Welcome to G Fun Facts Online. We are diving straight into a medical revolution that sounds like it was pulled directly from a science fiction novel. The syringe has long been the undeniable hero of public health. It is the tool that has eradicated diseases, managed global pandemics, and saved countless millions of lives. However, for all its life-saving power, the needle comes with profound limitations. It relies on strict, ultra-cold chain logistics. It suffers from prohibitive manufacturing bottlenecks. And let us not forget the widespread vaccine hesitancy, driven by a very basic, very human fear: injection phobia. Furthermore, while injectable vaccines are excellent at creating systemic immunity, meaning they build strong defenses within your bloodstream, they often struggle to mount defenses at the body's mucosal surfaces. These mucosal surfaces, like the lining of your nose, throat, and lungs, or your gastrointestinal tract, are the exact entry points for respiratory and enteric pathogens. Injecting a vaccine into a muscle is like putting all your guards inside the castle keep, leaving the outer walls completely undefended against invading armies. But what if the future of immunization did not involve a sterile clinic and a sharp jab? What if it simply involved swallowing a freeze-dried capsule, eating a snack, or even drinking a fermented beverage? Enter a radically different paradigm in medicine: edible vaccines. At the forefront of this biotechnology revolution is a microscopic fungus you are likely very familiar with: transgenic brewer's yeast, known scientifically as Saccharomyces cerevisiae, alongside its probiotic relatives like Saccharomyces boulardii, and industrial counterparts like Pichia pastoris, also known as Comagatiella phobiae. This humble fungus, historically responsible for human civilization's bread and beer, is undergoing a high-tech renaissance. Through advanced genetic engineering, yeast is being transformed into a programmable biological factory and delivery vehicle, capable of surviving the human digestive tract to deliver life-saving immunizations directly to your gut. To understand why this is such a monumental breakthrough, we first have to understand the biological armor of yeast and how it survives the incredibly hostile gut environment. The concept of an oral vaccine is historically challenging, because the human gastrointestinal tract is literally engineered by evolution to destroy complex biological molecules. Think about it. When you eat food, your digestive system breaks it down into basic biological building blocks. The human stomach is essentially a cauldron of hydrochloric acid with a highly corrosive pH, followed by intestines awash in bile salts and proteolytic enzymes designed to shred proteins apart. If you were to swallow a standard naked protein subunit vaccine, or one of the fragile mRNA lipid nanoparticles used in recent global vaccination campaigns, it would be completely obliterated within minutes in this acidic environment. This harsh physiological reality is exactly why the few oral vaccines available today generally rely on live attenuated pathogens, which are weakened viruses strong enough to survive the stomach, but too weak to cause full-blown disease. However, brewer's yeast is uniquely equipped for this perilous journey down the digestive tract. The yeast cell boasts a robust, complex outer wall composed primarily of thick layers of beta-glucans, mannans, and chitin. If you are familiar with chitin, it is the same tough material that makes up the exoskeletons of insects and crabs. This microscopic architectural marvel acts as an impenetrable biological vault. It completely shields the delicate, genetically engineered viral or bacterial antigens housed within the yeast cell or displayed upon its surface from acidic and enzymatic degradation. Consequently, the yeast guarantees safe passage for its precious vaccine cargo, straight through the stomach and directly into the lower intestine. And the lower intestine just happens to serve as the epicenter of the gut's immune system. But surviving the journey and delivering an antigen to the gut is only half the battle. Once there, the immune system must be provoked to recognize this antigen and respond to it. This brings us to how yeast acts as nature's syringe, perfectly engaging the gut's immune network. The human intestine is lined with an extensive and highly active network of immune cells known as gut-associated lymphoid tissue, or GALT. Within this tissue lie Peyer's patches, which are specialized lymphoid nodules covered by uniquely adapted cells called microfold cells, or M cells for short. Think of M cells as the border patrol agents of your gut. When the transgenic yeast arrives in the intestine, these M cells readily recognize, capture, and engulf the yeast particles. They then translocate them across the intestinal barrier to underlying antigen-presenting cells, such as macrophages and dendritic cells. Here is where a fascinating biological synergy occurs. The yeast cell wall does not merely act as a protective vehicle, it functions as a powerful, innate immune adjuvant. An adjuvant is a substance added to a vaccine to boost the immune response. Many traditional injections require controversial chemical adjuvants, like aluminum salts, to wake up the immune system. Yeast does not need this. The beta-glucans on the yeast surface bind directly to pattern recognition receptors, specifically ones known as DECTIN1, on the surface of macrophages. This sets off a targeted immunological alarm. The yeast naturally stimulates a robust immune response purely through its biological structure. Crucially, oral yeast-based vaccines have the unique ability to trigger a dual-layered immune response. First, they stimulate strong systemic immunity, creating circulating IgG antibodies in your blood. Second, and perhaps more importantly, they generate high levels of secretory immunoglobulin A, or SIGA, at mucosal sites. This mucosal immunity acts as that frontline barrier we talked about earlier. It neutralizes pathogens in the respiratory or gastrointestinal tracts before they can successfully cross the epithelial lining and establish an infection. You are stopping the invaders at the gates. Now, let us look at the biotechnology behind the brew. How exactly is a baker's microbe transformed into a prophylactic biotherapeutic? The magic lies in recombinant DNA technology. Scientists utilize several highly elegant strategies to arm yeast with pathogen-specific antigens. One of the most prominent and successful techniques is called yeast surface display, or YSD. Biotechnologists exploit the yeast's natural cell wall proteins. They most commonly utilize the A-agglutinin AGA1P AGA2P system. By using this system, scientists can genetically fuse a viral or bacterial antigen to these native anchor proteins. Imagine the yeast putting on a coat made of the virus-recognizable parts. As the yeast grows and divides, it literally wears the vaccine on its exterior, presenting the target antigen directly to the host's immune cells upon ingestion. Alternatively, researchers employ intracellular expression. This prompts the yeast to manufacture and accumulate antigens internally. In some cutting-edge designs, these internal proteins self-assemble into virus-like particles, or VLPs. These are hollow protein shells that mimic the precise three-dimensional architecture of a real virus, but they do not contain any infectious genetic material. It is a perfect molecular decoy. To ensure the genetic stability of these transgenic yeasts, modern bioengineering strongly favors permanent genome integration over traditional plasmid vectors. Let me explain why. Plasmids are circular loops of DNA used to insert new genes. However, these loops can be easily lost by the yeast during large-scale industrial fermentation unless they are maintained by adding antibiotics to the yeast's food source. Adding mass amounts of antibiotics to industrial vats is a practice heavily restricted by health regulators due to the global crisis of antibiotic resistance. So instead of loose plasmids, scientists use targeted tools like CRISPR-Cas9 or homologous recombination to integrate the antigen-producing gene directly into the yeast's chromosomes, such as chromosome IV. This creates highly stable yeast strains that can be cultivated in massive, antibiotic-free bioreactors. The versatility of this transgenic yeast platform has sparked a golden age of immunological research, showing immense promise across both human and veterinary medicine. Let us look at some frontline applications, starting with global pandemics and respiratory invaders. During the COVID-19 pandemic, the logistical nightmares of ultra-cold chain mRNA vaccines became glaringly apparent, especially in low and middle-income countries. Transporting vaccines at -70°C across deserts or remote rural areas is nearly impossible. In response, global researchers pivoted to yeast. Multiple candidate vaccines were rapidly developed using Saccharomyces cerevisiae and Pichia pastoris to express the SARS-CoV-2 spike receptor binding domain and nucleocapsid proteins. When administered orally to mice, these yeast vaccines elicited robust neutralizing antibodies and a mixed Th1-Th2 type cellular immune response, with a distinct Th1 bias, which is highly effective against viruses. But here is a fascinating twist. Oral administration of the SARS-CoV-2 yeast vaccine achieved an unexpected secondary therapeutic benefit: gut microbiota reconstruction. COVID-19 infections frequently cause severe gastrointestinal distress and dysbiosis, which is a harmful imbalance of the gut microbiome. Studies revealed that the yeast vaccine not only provided specific viral immunity, but also fostered a more complex, healthier gut flora in animal models. It was effectively acting as an immunoregulatory probiotic while simultaneously training the immune system to fight off the virus. Beyond human pandemics, veterinary medicine is currently the most active proving ground for edible yeast vaccines. The global agricultural sector faces staggering economic losses from infectious livestock diseases and is under immense legislative pressure to drastically reduce antibiotic usage. Transgenic yeast offers a highly scalable, feed-based solution that can be seamlessly integrated into animal diets. For example, researchers successfully engineered yeast to express the capsid protein VP2 of the infectious bursal disease virus, a highly contagious, immunosuppressive poultry disease. When given orally to chickens, it induced specific antibodies and protective mucosal immunity without requiring a single individual injection. Imagine trying to inject thousands of chickens one by one. Now imagine just mixing the vaccine into their feed. The efficiency is unparalleled. Similarly, to combat pylorem disease, a devastating illness in broiler chickens caused by Salmonella pylorum, scientists bioengineered yeast to display the bacteria's outer membrane and fimbriae proteins. A pivotal 2026 study demonstrated that this orally administered vaccine increased chick survival rates and drastically reduced Salmonella loads in their muscle and tissues. This is a massive win for the vital One Health ecosystem approach, which recognizes that the health of people is closely connected to the health of animals and our shared environment. In the swine industry, multi-drug-resistant F4-positive enterotoxigenic Escherichia coli, or ETEC, causes fatal post-weaning diarrhea in piglets. This represents a massive threat to animal welfare and global food security. A recent breakthrough utilized a recombinant yeast strain displaying the bacterial FAEG protein. Fed directly to the animals, the vaccine significantly enhanced intestinal health, upregulated mucosal immune factors including interleukin-2, interleukin-4, and interferon gamma, and provided crucial, life-saving protection against ETEC infection. And the technology is continuously being adapted for challenging human, enteric, and viral diseases. Researchers at institutions like the Vilnius University Life Sciences Center are aggressively exploring yeast-based oral vaccines for polyomaviruses using yeast that intracellularly generates major capsid proteins to form those virus-like particles we discussed earlier. Furthermore, groundbreaking studies are utilizing yeast-derived beta-glucan microparticles to deliver DNA and mRNA directly to macrophages. This hints at a near future where even highly complex genetic therapies could be administered orally, entirely circumventing the rapid degradation that usually plagues mRNA in the gastrointestinal tract. This brings us to one of the most attractive attributes of yeast-based vaccines, moving beyond the pill into lyophilization and the concept of the beer vaccine. The remarkable physical resilience of yeast liberates global health initiatives from the tyranny of the cold chain. Traditional vaccines require constant, uninterrupted refrigeration. That is an infrastructural hurdle that severely limits access in remote, rural, or developing regions. Transgenic yeast, conversely, can be lyophilized, which means freeze-dried, into stable powders, capsules, or even edible crisps that endure high temperatures and boast an exceptionally long shelf life. Because Saccharomyces cerevisiae holds a GRAS status, which stands for generally recognized as safe in the food industry, the potential delivery formats border on science fiction. Experiments have already shown that animal models willingly consume dried yeast vaccine preparations as part of their normal diet. For human applications, while pharmaceutical-grade clinical capsules offer the most precise dosing, researchers have successfully demonstrated the proof of principle for integrating vaccine antigen-producing yeast into everyday fermented beverages. Virologists have colloquially dubbed this the beer vaccine. They are actively exploring whether probiotic drinks or non-alcoholic fermented beverages like kvass could theoretically double as highly accessible, population-level immunization tools. Just imagine grabbing a fermented drink from a local shelf-stable pantry to receive your annual booster. Despite its revolutionary potential, the journey of edible yeast vaccines from the laboratory bench to the local pharmacy is fraught with both biological and bureaucratic challenges. The first major biological hurdle is navigating oral tolerance. The human digestive tract is evolutionarily designed to tolerate vast quantities of foreign dietary proteins without launching a systemic inflammatory immune attack. If it didn't, we would have severe allergic reactions to every meal. A successful oral vaccine must strike a highly delicate balance. It must be sufficiently immunogenic to break this default tolerance and provoke active immunity, but not so aggressive that it triggers runaway mucosal inflammation or severe food allergies. Regulatory frameworks pose an equally daunting obstacle. A live, transgenic yeast cell is, by legal definition, a genetically modified organism, or GMO. While the medical use of GMOs is widely accepted in closed manufacturing environments, such as the massive steel vats used to produce synthetic human insulin, the intentional release of a live, replicating GMO directly into the human digestive system and subsequently into the environment via wastewater triggers incredibly stringent environmental and safety regulations. Simplifying and standardizing these complex legislative pathways for advanced therapy medicinal products will be absolutely critical before large-scale human clinical trials can proceed unhindered. Furthermore, standardizing dosage is inherently complex. Unlike a sterile injection, which delivers a precise quantified microgram measurement of an antigen directly into muscle tissue, the surviving payload of an oral yeast vaccine can vary drastically from patient to patient. Vaccine efficacy can be heavily influenced by an individual's unique stomach acid pH, their pre-existing gut microbiome composition, and their intestinal transit time. Establishing reliable pharmacokinetic models for a living, digesting microbe remains a primary focus of ongoing clinical research. In conclusion, the future of edible biotherapeutics is incredibly bright. The concept of swallowing a vaccine, rather than receiving an injection, represents a profound paradigm shift in preventative medicine. Transgenic Brewer's yeast has evolved from a simple agent of culinary fermentation into a highly sophisticated programmable nanobot. It is capable of surviving the biological gauntlet of the human gut, naturally stimulating the mucosal immune system and delivering life-saving antigens right where they are most effective. As biotechnology research rapidly advances into cutting-edge areas like glycan humanization, where yeast sugars are modified to look more human to prevent immune rejection, alongside thermostable formulations and highly localized mucosal delivery, yeast platforms stand ready to democratize global health. They promise a future where pandemic preparedness does not rely on acquiring expensive ultra-cold freezers and sterile syringes, but rather on highly scalable, decentralized bioreactors capable of producing low-cost, shelf-stable, and entirely edible immunizations. The exact same humble microbe that helped ancient civilizations bake bread and brew beer is right now being drafted to fortify the human immune system against the pathogens of tomorrow. Thank you for listening to this deep dive on G Fun Facts Online.